UCLA Tarjan Center. On behalf of the Tarjan Center faculty and staff, it's my pleasure to welcome you to our distinguished lecture series and to welcome our distinguished speaker, Dr. Carrie Bearden. Now, by way of background, Dr. Bearden received her PhD in clinical psychology from the University of Pennsylvania and joined the UCLA faculty in 2003. Now, currently, she is a professor in the Semel Institute for Neuroscience and Human Behavior and also the UCLA Brain Research Institute, and has joint appointments in the Department of Psychology at UCLA. Dr. Bearden's research aims to understand neurobiological risk factors for the development of serious mental illness in youth using methods to study cognition and neuroanatomy in clinical high-risk groups. And this includes adolescents with early symptoms of psychosis and in highly penetrant genetic conditions. Her recent work focuses on translational approaches to understanding disrupted brain circuitry in developmental neuropsychiatric disorders, particularly in the context of rare genetic disorders. Among her numerous titles, Dr. Bearden is the director for the UCLA Center for Assessment and Prevention of Prodromal States, also known as CAPS, and she co-directs the Neurobehavioral Genetics Training Grant at UCLA and is an assistant editor of Biological Psychiatry, Cognitive Neuroscience and Neuroimaging, the Schizophrenia Bulletin, and Biological Psychiatry. So Dr. Bearden is a fellow of the American College of Neuropsychopharmacology and the Association for Psychological Science. She also chairs the DSM-5 Serious Mental Disorders Committee and the Women's Leadership Group of the Society of Biological Psychiatry. So she has many very distinguished titles. Dr. Bearden has received numerous awards and honors, both for her research achievements and also for her teaching and her mentorship. So today, Dr. Bearden will be sharing her really innovative and important work related to a genetics-first approach to autism and psychosis spectrum disorders. We encourage you all to ask questions throughout the lecture. You can use the Q&A feature on the Zoom toolbar, and our Director of Training, Dr. Jasper Estebio, will be moderating those questions at the end of the talk. But in the meantime, and without further ado, please join me in giving a warm virtual welcome to Dr. Carrie Bearden.
Thank you so much, Liz. Thank you for that really wonderful introduction. I hope you can all hear me okay. Let me know if not. But I want to thank Dr. Laugesen and Dr. Estevio for inviting me to speak here for this lecture series. It's really an honor to be here. So, and like Dr. Laugesen said, please feel free to ask questions throughout, and I'll definitely try to leave some time at the end for discussion. So here's a lovely picture of UCLA, and I recently switched my acknowledgement slide, my thank you slide for everyone in my lab and my team who has done all this hard work. I wanted to make sure to put that at the very beginning so that to give the proper credit and not shortchange the folks who actually did all this hard work that I'm going to be speaking about. So thank you, everyone. It's obviously a village, a global village to do this kind of work. All right, so let's talk a little bit about what we know about the genetic architecture of psychiatric disorders. So recently, there's been major progress in terms of connecting genes to psychiatric disorder phenotypes, particularly for schizophrenia, also several other developmental neuropsychiatric disorders. Finally, in these really very large sample sizes, there are hundreds of genome-wide significant risk variants. But that being said, we still have a very poor understanding of mechanism. That is to say, our mechanistic understanding really continues to lag behind the pace of gene discovery. So how do we get from these genetic risk loci that constitute these diagnostic categories? This is a very complex system, and there are many intermediate traits that underlie the development of these disorders. And each of these genes, and this is a Manhattan plot from a genome-wide association study, so each of these genes we know individually contributes a very small degree of risk, possibly in combination with environmental factors can cause someone to reach the threshold for developing an actual disorder. But this is a very hard problem, particularly given the remarkable complexity and heterogeneity of these disorders. And so one approach and one possible way forward is to examine these what we call pathophysiologic processes, the level of neural circuits, systems, cognition, and behavior that mediate between this genotype and this distal disease phenotype. And at the same time, in addition to these common variants over here that each is contributing a small degree of risk, we also have these rare genomic disorders, highly penetrant for neuropsychiatric disorders. They confer substantially elevated risk, and they may therefore represent a specific, more homogeneous subtype with a well-characterized genetic etiology that can potentially, we hope, shed light on disease mechanisms in the broader population. And some of the advantages of studying with this, taking this genetics first approach, is that it allows the development of animal models, so really doing these translational studies that have strong construct validity. So you're studying the same genetic defect in an animal in a mouse model, as opposed to, trying to convince yourself that a mouse has schizophrenia. And they also present these a system to dissect the circuitry mediating behaviors. And the other thing that's really nice and important for prospective longitudinal studies, which is something that we are very interested in, is that they can be identified very early in development, possibly even in utero. And so what I'm going to be speaking about today is particularly the 22q11.2 microdeletion syndrome, and I'll get a little bit into the duplication as well. Duplication of the same genomic locus. And these disorders as particular models for understanding neuropsychiatric disorders. So, as I mentioned, there's been a lot of progress in our understanding of the role of common variants to neuropsychiatric disorders. There's also been major advances in our understanding of the contribution of rare variants to the genetic architecture of these disorders, most notably autism spectrum disorder and schizophrenia. So while the majority of cases of developmental neuropsychiatric disorders remain of unknown or idiopathic etiology, these rare genomic copy number variants, these deletions or duplications of genomic material, may account for a larger proportion of cases than was previously believed. So at present, we think about 2% of cases of schizophrenia and at least 25% of cases of simplex autism spectrum disorder. So like one case of autism in a family in particular. And those numbers keep going up actually, and more with an identified genetic etiology. And the fact that we see these rare variants where you have one disorder that can be caused, like schizophrenia, that can be caused by multiple different mutations, as well as the converse, you have one mutation that can lead to multiple different disorders. This really provides evidence for genetic pleiotropy and also challenges these widely held views that we have of diagnostic categories. So the fact that we do have these rare disease models of complex neuropsychiatric disorders really gives us an opportunity to take what we'd call a genetics first approach. And this was really nicely outlined in a paper from the Simons VIP Consortium. And the logic behind this approach is based on the increasing evidence that genetics underlying neuropsychiatric disorders are complex and may involve mutations in hundreds of genes, each of which is relatively infrequent. And so this heterogeneity makes it quite challenging to perform patient cohort studies because there may be characteristics that are specific to certain subsets that are not common to all individuals. But the fact that we do see these certain highly penetrant copy number variants, genetic variants, potentially even in single genes that are observed recurrently in cohorts ascertained by psychiatric diagnosis gives us the potential to accelerate the shift towards personalized therapeutic approaches and to contribute new insights. So this is a really important question, and it has certain treatment implications. So are treatments best tailored to a specific genetic event, or might they be generalizable to those with a neurodevelopmental or psychiatric illness of other or unknown etiology? So these are some of the questions that we're really been very interested in. And so I'm going to be talking particularly about the 22q11.2 microdeletion syndrome. This is a genetic syndrome that I got really interested in actually when I was a graduate student at the University of Pennsylvania. And there's a very large center at the Children's Hospital of Philadelphia. And I was seeing a lot of these children clinically. So it has a pretty long history. It's one of the first recurrent copy number variants to be discovered. Has a also known as villocardiofacial or DeGeorge syndrome. While it's a rare disorder, it's still relatively common. It's about 1 in 4,000 live births. And so it results from this hemizygous that's deleted on one copy of your 2 chromosomes 22 and on the Q arm. And most patients, about 85%, have a similar about 2.6 megabase deletion. So it's a large deletion. There are some associated physical features, cardiac defects, immune deficiency, craniofacial anomalies, as well as intellectual disability. We got particularly interested in it because of this really high rate of schizophrenia. It's quite remarkable, about 25 times the general population risk. But there's also, well, and this is from the Psychiatric Genomics Consortium, just showing that genome-wide, this 22q11.2 deletion really carries the strongest risk of schizophrenia across these various copy number variants. But there's also what I would call a lot of diagnostic pluripotency in childhood. There's also quite elevated rates of autism spectrum disorder, quite variable across studies, as well as ADHD and anxiety disorder, which are really quite prevalent diagnoses. So some of the key questions that we've been really interested in, I mean, first of all, as a starting point, there's really limited data on individuals that are carrying the same highly penetrant copy number variant. And so we're interested, first of all, in risk factors for psychosis in a high penetrance genetic variant like the 22q deletion syndrome, and really whether these risk factors overlap with what we see in idiopathic or behaviorally defined illness. So related question, are there consistent and reproducible effects of the 22q deletion on brain structure? And do these effects converge with those observed in idiopathic psychotic disorder? What are the mechanisms underlying these diverse phenotypic presentations that we see in the 22q deletion syndrome? And then also gene dosage effects may provide some really important clues into disease biology. So we're interested in what are the neurofunctional consequences of these reciprocal imbalances of having a deletion or a duplication at this locus. And an important premise for this work is that copy number variation, having a segment of DNA that's deleted or duplicated, may have a more powerful effect on intermediate phenotypes than does common genetic variation. And so I'm going to show you a little bit of work from our International Brain Behavior Consortium. This was work led by Maud Schneider, and this was a combined effort across 22 sites as part of this consortium. And so across sites, we were able to accumulate a total of 1,402 individuals with a molecularly confirmed 22q11.2 deletion. And here we were looking specifically at the rates of schizophrenia spectrum disorders across the lifespan by age. And what you can see, and so the schizophrenia diagnosis is shown in blue. And so even in young children, there's not very many of them, but there are still some. Majority are what would be called psychosis not otherwise specified, or atypical psychosis. But you can see that schizophrenia diagnosis really does tend to crystallize over the lifespan. And in fact, in this combined cohort, psychotic disorders were present in 41% of the adults over age 25, which is really quite remarkable. So as I mentioned, schizophrenia is, well, it may be the most specific diagnosis that's probably gotten the most study. There are a lot of other comorbidities in the context of this deletion syndrome. And so we see that there is affective dysregulation is a really prominent feature. I mentioned also rates of autism spectrum disorder, anxiety, and ADHD being some of the most common diagnoses. And comorbidity rates we found in the literature are really not frequently reported, but it is the rule rather than the exception. And this is true not just of the 22q deletion, but if you look across at other rare genomic disorders, this really tends to be the case. And transdiagnostically, we do see this affective dysregulation. And so we tried to do, this is a review paper from a few years ago, just looking at the rates of various psychiatric diagnoses across the lifespan. And so you can see that early in childhood and adolescence, very high rates of ADHD, still gets a little bit less over the lifespan. Psychotic disorders, we really see tends to increase in adolescence and young adulthood, consistent with the general population. Anxiety disorder is very prevalent throughout the lifespan, and then as well as autism spectrum disorder. So turning now to some of the risk factors for psychosis that we see. This is from a general population study, not of 22q deletion, but this was an epidemiologic study, the Dunedin cohort in New Zealand. And they were interested in cognitive decline, so a drop in IQ as a predictor of psychosis. And so what they saw here in this general population study is that IQ tends to be quite stable over the lifespan between the ages of 7 and 38 in healthy people, in people who develop depression, and in individuals with mild cognitive impairment. It's lower but stable, whereas in those who develop schizophrenia in adulthood and young adulthood, there was a pretty substantial drop in IQ, almost 0.4 standard deviations between the ages of 13 and 38. So we were really curious about whether we would see a similar pattern in people with 22q deletions who developed schizophrenia. So again, this was in our multi-site international brain behavior cohort. We had 430 individuals with IQ assessed at least 2 time points. And so what we're looking at here is the average verbal IQ change per year. The red is subjects who did not develop a psychotic disorder, and the green is in those who did. And so you can see that compared to the general population, even in those that don't develop psychosis, there's still a relative decline in IQ. And but that decline is substantially steeper. This is a significant difference in slope. So significantly greater decline in those who develop psychosis. And this shaded red line here is the mean age of onset of psychotic disorder in this cohort. So you can see that the rate of cognitive decline was happening many years before the onset of psychotic disorder, pre-pubertally between the ages of 8 and 12. And this is an odds ratio of 3.5. So it's a pretty robust predictor of subsequent psychotic illness. Now, that's not the only risk factor for psychosis in this population. There's a number of others, some of which have also been studied in our international brain behavior cohort. So inattention symptoms is one that has been shown in idiopathic or behaviorally defined psychosis to be a pretty potent risk factor. And so we found that to be the case here as well. Dimensionally measured inattention symptoms, as well as the categorical diagnosis of ADHD, that was even more of a potent risk factor, particularly if it was diagnosed at 2 time points. That's an odds ratio of almost 10 for predicting a subsequent psychotic symptom development. And then, and that's for psychotic symptoms, things like delusions, hallucinations, and then ADHD diagnosis at the 2 time points was also associated with development of an overt psychotic disorder like schizophrenia later on. Now, there are a number of other predictors. Some of these have been looked at only in the context of pretty small studies, like negative symptoms, things like anhedonia, amotivation, as well as functional impairment. So a lower score of global assessment of functioning score. Those were both found in a single site study to predict the emergence of psychotic spectrum symptomatology, as well as interestingly, impairment in odor discrimination. And this is an interesting one because this has also been seen in behaviorally defined schizophrenia to predict illness onset. Inflammation is another risk factor that is certainly interesting because high circulatory levels of pro-inflammatory cytokines have been associated with schizophrenia in the general population. And because of the immune deficiency in 22q11, there's been a lot of interest in investigating this. Again, these are pretty small studies so far, but it certainly, there are some suggestions. This is a risk factor. Now, it's also important to point to things that don't predict schizophrenia that aren't risk factors. And in fact, in a prospective longitudinal study, there was no association between a childhood diagnosis of autism spectrum disorder in 22q and subsequent development of psychotic disorder. So these are both pretty prevalent, but they don't appear to be linked to each other. Now, I mentioned earlier on, if you can remember a couple of slides ago, that the rates of autism spectrum disorder in this population were really variable, ranging from 12 to 50% across studies. And so this is something that we were interested in. We know that these core autism behaviors, things like impairment in joint attention, gestural communication, initiating conversation, restricted interest, as well as language delay. Language delay is actually present in probably close to 100% of kids with 22q deletions. So these core autism behaviors are pretty characteristic, but certainly not all of them meet criteria for a diagnosis. So we were interested in whether there are distinct biological substrates associated with the categorical autism spectrum diagnosis, or whether a dimensional approach, looking at this as a severity continuum, better fits the pattern of findings. And so this is from our UCLA cohort. We had about 60 or so, more than 60 participants. 29 met criteria based on the ADI and the ADOS for an autism spectrum diagnosis. 32 did not. And we had 55 demographically matched controls, matched in terms of age, sex, and education level. You'll note that IQ was significantly higher in the controls, but did not significantly differ between those with and without an autism diagnosis. And so both, as I just mentioned, both 22q11 cases with and without autism spectrum disorder diagnoses had significantly lower full-scale IQ, but they didn't differ between each other. Now, we did see, and this is shown here, these were z-scored to the controls. So the controls are this line here. And what we're looking at is the degree of impairment across these measures in those with and without an autism diagnosis. The light red was without, and the darker red is those with. And so there are a couple of neurocognitive domains when we define autism categorically, significantly poorer in those with an autism spectrum diagnosis. The processing speed, the back symbol coding, as well as measures of face memory, significantly worse in those face memory as well as a spatial memory task. They actually didn't differ in terms of emotion identification and theory of mind tasks, so these kind of classic social cognition, but both groups were fairly impaired relative to controls. Now, in terms of neuroanatomy, we also looked at some neuroanatomic measures, and we found that those with the autism diagnosis had significantly lower amygdala volume, the right amygdala specifically, as well as differences in parahippocampal cortex, the thickness of that cortex, significantly thinner in those with an autism diagnosis. And so what we were interested in doing was to see whether it was a dimensional or categorical fit, but better fit the data. And so we used a measure called Akaki's criterion, which is a commonly used measure for model selection. And what we found was not that straightforward. Basically, it's complicated. We found that a dimensional model was the best fit for cognitive measures and for amygdala volume, but in fact, a categorical model, so autism, yes, no, was a better fit for this parahippocampal thickness. So this is really, and I'd be happy to discuss this more in the discussion. I think this is something that requires further investigation, but essentially, there's value to defining autism both categorically and dimensionally in the context of these rare genomic disorders. So I'm going to turn now to a little bit of our brain imaging work. And I realize that for some audiences, some people are really, really interested in brains and some people not so much. So bear with me. Hopefully you'll find this interesting. This is work from our another multi-site consortium called the ENIGMA consortium. It stands for Enhancing Neuroimaging Genetics Through Meta-Analysis. And so essentially, this was well, now multiple consortia, but was started by my colleague Paul Thompson when he was at UCLA, and essentially trying to combine data from anyone in the world with a DNA sample and a brain scan. And so this kind of data pooling, this kind of team science approach really allowed us to identify the first genetic variants that were associated with human brain structure. And so this ENIGMA consortium has now really grown, it's exploded, and diversified into, I think, over 300 different working groups. So I'm really excited to be leading the 22q working group, and we have a number of sites around the world. And this, again, really allowed us, thanks to the generosity of all of these folks involved, to have a pretty large sample, 537 with a brain imaging, with a 22q deletion, 331 age and sex match controls, as well as some individuals with the 22q duplication, which I'll get to shortly. So let me give you a very brief overview of methodology and how these brain imaging indices, how we think they map to biology. So overall volume of brain structures is actually the product of two neuroanatomic features, that is cortical surface area, the total area covered by the cortex in a given region, and cortical thickness, which is the average thickness of 6 cortical layers within a region. And so these indices really appear to be driven by distinct genetic mechanisms. They also have different developmental trajectories, so it's important to look at them separately. Surface area, we know to be impacted by regulation of neural progenitor proliferation and cell cycle in the developing brain. And cortical thickness is shaped by later developmental processes, possibly synaptic density and myelination processes. So that I think is an important distinction, and which it'll become important later. So in terms of our methodology, we used a method called free surfer, pretty standard processing pipeline, and we covary for age squared to get at nonlinear effects of age, as well as sex and site. And I'm happy to talk more about methods for anyone who's interested. This is work led by Frank Sun, who is an incredible psychiatrist who does a lot of brain imaging in his spare time. And so essentially what we're looking at here are brain maps of looking at the differences in cortical thickness, shown here on the left, between 22q deletion cases and controls. And the blue is showing that 22q11 patients actually had relatively increased cortical thickness, with the exception of these specific areas, the superior temporal gyrus, which is critical for language, and the anterior cingulate here, which is brain structure critical for attention and motivation. Now, surface area looks really different. We see this very pervasive and widespread reductions of cortical surface area in 22q deletion cases. And these patterns were pretty dramatic in terms of the effect sizes. And so Frank did a machine learning algorithm to see whether we could classify cases versus controls based on these brain imaging patterns. And it does a really good job with 125 cortical features. We were able to classify based at 94% accuracy. And what's really interesting is when you compare that to idiopathic or behaviorally defined disorders like bipolar disorder, you get up to a maximum of about 65% accuracy for classification. So it just really speaks to the homogeneity and the consistency of the neuroanatomic signature of this disorder. And in fact, if you just took 2 cortical features, the cuneus and the lingual gyrus surface area, you can still get, you see this clustering here, you still get about over 86% accuracy in classification. Now, given the high rates of psychosis in this population, we really wanted to see whether there was a similarity between the brain imaging patterns we see in idiopathic or behaviorally defined schizophrenia and 22q. And so this is just comparing 60 and 60 demographically very well matched cases with 22q with and without a psychotic disorder. And so here, the pattern's quite different. We see this, it's particularly cortical thinning, or the thickness of the cortex that's different. And particularly in these medial frontal regions, slightly in temporal cortex as well. So what we call Hereromodal association cortex. And this looks actually very similar to what we see in idiopathic behaviorally defined schizophrenia. And if we do a quantitative comparison, there was significant convergence between the brain regions most affected in 22q associated psychosis and behaviorally defined schizophrenia. But this was but we did not see an overlap with brain regions most affected in major depression. So it did appear to be relatively specific to schizophrenia. Now, how about at the level of subcortical structures? So we're talking about the cortex. And so this was work led by Chris Ching, who's a really incredible neuroscience graduate student, now a PhD, I'm sorry, now a postdoctoral fellow at USC. And so what Chris found was that overall, we see these really significant, particularly in the hippocampus, as well as some other subcortical structures, but increases in striatal regions like the nucleus accumbens in 22q deletion cases. And Chris applied this really very cool, novel subcortical shape analysis. And what you can see here is that this overall volumetric difference was really sort of masked by this complex pattern of increases and decreases across these subcortical structures. And in particular, we saw that there were subregions with projections to frontal cingulate and association cortices were particularly affected. Whereas when we look, and this was for 22q overall, but when we compared those with and without a psychotic disorder, we see these prominent reductions in the hippocampus, the amygdala, and the ventral thalamus in these 22q cases with psychosis compared to those without. And again, comparing that to idiopathic or behaviorally defined schizophrenia, we see a very high correlation between the brain structures that were most affected in 22q psychosis and idiopathic psychosis. Now, in contrast to the cortex, we see actually that this pattern looked very similar compared to other neuropsychiatric disorders as well. So 22q associated psychosis had very similar patterns of subcortical deficits compared to major depression, as well as obsessive compulsive disorder and bipolar disorder. Where we did not see a similarity, though, was in ADHD and autism spectrum disorder, behaviorally defined. So those patterns look quite different from 22q associated psychosis. Now, this brings us to the question of what are the genes in this locus that are driving these brain phenotypes? So we know that this is what we call a genomic hotspot region. The region is very prone to mistakes in recombination due to these segmental duplications that are flanking the region. And so the typical deletion is what we spans these A to D breakpoints. As I mentioned, it's a big region. It's 46 protein coding genes in the region. Several of these genes are highly expressed in the brain, involved in early neuronal migration, cell fate decision in the cortex, and also what's become really interesting recently is microRNAs, the role of microRNAs in the region. These are these small non-coding regulatory RNA molecules that contribute to basic mechanisms underlying brain development and plasticity. So before we even get to specific genes, let's just talk about the effect of the deletion size. So about 85% have this similar large deletion, but about 8 to 10% have a smaller, which is typically 1.5 megabase deletion. And so again, in the context of our international brain behavior study, we looked at differences between those of the smaller versus larger deletion. And we found that full-scale verbal and performance IQ significantly higher in those with the small deletion. So these IQ metrics were decreased by 0.3 to half a standard deviation in those with the larger versus smaller A to B deletion. And so we were really interested in whether brain structural differences might underlie the effect of deletion size on IQ. And our subcortical shape analysis revealed these local differences as a function of deletion size. So we actually did not detect with this gross volume analysis. So significantly greater alterations in subcortical brain structures in those with the smaller deletion. And we also see this in the cortex where there was significantly greater cortical surface area in those with a smaller deletion. So in other words, having a larger deletion is going to have a more severe impact on brain and behavior. Not particularly surprising, but this was the first time that any study really could amass enough subjects to really investigate this question. So this was exciting. So I mentioned, I think in the beginning, I was going to talk a little bit about 22q duplications. So this was a study that came out, I believe in 2014, evidence that duplications of 22q protect against schizophrenia. This was from the group at Cardiff, which was an amazing headline, really, because this would be really the first putative protective mutation against schizophrenia. And so essentially what they found was that in their discovery cohort, there was a significantly lower rate of duplication of 22q duplications in schizophrenia cases than in the general population. And then they had a replication sample as well. So again, you have a lower risk of schizophrenia if you have a 22q duplication than the general population does. And this sounds really remarkable, but in fact, this has been replicated a couple of times now, including in a Han Chinese population. So really quite fascinating. And this is something we're really, really trying to understand what this potential mechanism could be. Now, in the context of other copy number variants, other reciprocal deletions and duplications of the same segment of DNA, interestingly enough, they have been associated with mirror image phenotypes. So this has been found for 1q21, where a deletion or duplication is associated with either micro or macrocephaly, smaller or larger brain. And at 16p11, these mirror image phenotypes actually affect body mass index. So you could have, so those with the 16p deletion actually have increased body mass index and increased head circumference compared to those with the duplication, where both of those are reduced. And this was studied in more detail, the brain structure and the Simon's VIP consortium, where they found that opposing this global opposing brain differences, which differentially affected cortical surface area with increases, let's see, hopefully I said this right, increased cortical surface area in the deletion group and decreased surface area in the duplication carriers, corresponding to these overall body mass index differences. So really interesting, and this really had not been investigated in the 22q duplication. The 22q duplication was described, the study came out in 2009, describing it as a new chromosomal syndrome. Now, interestingly, in comparison to the deletion, which tends to be de novo, it's a new mutation, the duplication tends to be familial, so most have inherited it from a parent. Now, a parent who has the duplication may have a normal or near normal phenotype, even though the genomic alteration is identical in the child, and the child may have obvious clinical features. So most commonly, I mentioned the duplication may be protective against schizophrenia, but it's certainly not protective against other types of developmental delays. These children do tend to have pretty high rates of autism spectrum disorder, developmental delay, language impairment. And so this was Stefan Sanders' study from the Simons Foundation, finding that elevated rates of both the 22q deletion and duplication in simplex autism cases. So there's an estimated frequency of 0.05% of this 22q11 duplication in non-syndromic simplex autism spectrum disorder. And so we were really interested in, we did an exploratory study here at UCLA, recruiting 22q duplication carriers in addition to our deletion sample. And what we have found so far, and this is an ongoing study, but while we thought these folks would be pretty rare, harder to recruit, we've actually gotten a lot of interest and found quite a few. So the study, so at the time we did this analysis, we had 38 in the duplication group and a pretty wide age range. But the groups were matched in terms of parental education, age, gender, and you'll note that there was a pretty high rate of psychosis in the deletion carriers, none in the duplication, and ADHD and autism spectrum disorder were quite high in both CNV carrier groups, but didn't differ from each other. And this is work led by Amy Lynn, who is a wonderful neuroscience PhD student who recently graduated. And so what we're looking at here is gene dosage effects on the 22q behavioral phenotype. So this red here are controls. So these are all Z-scored to controls. So anything down here means their performance was impaired compared to controls. They were all scored the same direction. So what you can see is that IQ was significantly more impacted in those with the deletion versus the duplication, but some cognitive domains like verbal memory and processing speed were pretty similarly impaired in these CNV carriers. Social cognition also somewhat worse in the deletion carriers. And then autism spectrum traits we also saw were elevated in both groups, whereas we saw a divergence in schizophrenia traits, both positive and negative symptoms, significantly higher, more elevated scores in those the deletion compared to the duplication. Now, I mentioned that these autism traits were not significantly different between groups, but when we parsed them at a more detailed level, particularly for looking across these dimensional measures, and particularly for repetitive behavior, we did actually see a difference. So we know that a diagnosis of autism spectrum disorder is a highly multidimensional final endpoint, which can be arrived at many different ways. And so looking at these more fine-grained distinctions of specific behaviors on the repetitive behavior scale, we saw specifically that it was stereotypies were significantly more elevated in those with the duplication compared to those with the deletion. And we looked at this a number of different ways, and including with the machine learning approach. And this really does tend to hold up that this stereotypies were significantly greater in the duplication versus deletion groups. Certainly requires replication. And then at the level of brain structure, we saw these circumscribed differences in cortical thickness, which were in opposite directions. So where the duplications had, people with the duplication had increased cortical thickness, we saw, where they had decreased thickness, we saw relative increases in cortical thickness in the deletion patients. And then the blue is cortical surface area, where we saw these really widespread differences. And again, we saw a pretty substantial decreased cortical surface area in the deletion subjects, relatively increased compared to controls in the duplication subjects. So, and the controls were having showing an intermediate pattern. So this is essentially what we found across the board in the brain. And when we look at the brain structures that were most affected across groups, and these are, again, Z-scored to the controls, the superior frontal gyrus, the precuneus, a supramarginal gyrus, and frontal pole, cingulate gyrus, bank of the superior temporal sulcus. These are brain regions that are really critically implicated in social cognition, social cognitive neural circuitry. And so there does appear to be a differential impact on brain structures involved in social cognition in these 22q copy number variants. So I don't know if we have any genetic counselors in the audience, but this is, I think, a really, if we do, this is, or even if not, I think this is a really helpful analogy. And this is from Janine Austin, who does a lot of genetic counseling related to neuropsychiatric disorders in not just 22q11, but other rare genomic disorders. And so one interesting finding is that, you know, while the 22q deletion is associated with a pretty high rate of medical comorbidities, cardiac defects, and so forth, as I mentioned, for many parents, the risk of psychiatric illness is really the main concern regarding their child's health. And this was, I think, a really helpful analogy that Janine Austin proposed with mental illness jars, where you have the 22q deletion. There's this big ping pong ball in the jar. So it's really this major risk factor, but then there are these other little ones, and the blue is these environmental risk factors. And you can increase the size of the jar here by adding these protective factors, things like social support, therapy, and so forth. But then these other little balls we could think about as potentially, other genetic factors, other environmental factors, or, other things impacting risk. But this is, I think, a helpful way of thinking about this. Now, I mentioned a few times that 22q11 is certainly not the only genetic locus associated with the high risk for neuropsychiatric disorders, schizophrenia and autism spectrum disorder in particular. And so in collaboration with a number of colleagues, Sebastian Jacquin at Montreal in particular, and we've recently started investigating distinct and common brain imaging patterns across multiple neuropsychiatric disorders, copy number variants. And so we're really interested in, what are these fundamental principles of how these genetic variations can affect brain architecture. And it's also notable that there may be particular neuropsychiatric profiles in subgroups as a function of the genetic etiology. The other thing that I think is worth pointing out is that at the level, particularly, certainly the level of brain structure, we do see substantially larger effect sizes on phenotype of these rare genomic disorders than we do for a behaviorally defined disorder. So this is just an example here of cortical surface area where we see that, and these are Cohen's d effect sizes. So those with behaviorally defined disorders, the effect size is much smaller than what we see for any of these rare genomic disorders that are considered neuropsychiatric CNVs. And so to visualize the profiles of these, were basically looking at 16p11 and 22q11 deletions and duplications that all have a pretty high rate of autism spectrum disorder associated. Sam Chauner, as a colleague who was interested in looking across these different CNVs and whether they had different profiles compared to heterogeneous autism spectrum disorder cohort. And in fact, we do see pretty substantial differences in the profile. So these are Z-scored to will be considered, this idiopathic autism spectrum disorder cohort. And so we saw these profile differences between genetic variant groups. And also importantly, 54% of the CNV carriers who did not meet the full diagnostic criteria for autism spectrum disorder still had quite elevated levels of autistic traits. So this is really, I think, clinically very important, that even if they're not meeting the diagnostic criteria, there's still a lot of these traits measured on a continuum going on. In the last couple minutes, I'm just going to talk about areas needing lots more attention, things we really don't know. So we think there's no reason to think that interventions used in idiopathic disorders for these conditions, cognitive behavioral therapy, social skills training, there's no reason to think that they would not be effective, but there's not a lot of evidence to support that. So this was from a recent review, starting point with hundreds of studies, and they found that there are really only 3 small studies that had looked at psychosocial interventions in 22q deletion syndrome, and they were all pretty small, mostly with a waitlist control. So really no randomized control studies. One was a small group social cognitive training. So these studies really did provide preliminary evidence for feasibility and effectiveness of these interventions, but it certainly requires a lot more study. Environmental risk factors, and I think this is also because this is with a genetic disorder, people have been studying the genetics, but not so much, what are the environmental risk factors, family environment, things like social support as a protective factor, socioeconomic status. So we certainly would think that they might have similar impacts to how they would affect people without a rare genomic disorder. But again, this requires further study. And also, these 22q11 disorders in diverse non-European populations are extremely understudied. And so these are all things we're trying to address as much as we can. Certainly not addressing all of these questions, but we have a new network called the Genes to Mental Health Network. This is an international collaboration funded by the NIMH and the National Institute for Child Health and Human Development. And so it's a consortium across North America, Europe, and Africa. And, we note, this is from our website, rare genomic disorders affect less than 1 in 2,000 people in the general population individually, but collectively, they're a major cause of developmental and psychiatric conditions. And so we're all working together, and noting that most of these rare variants have been studied separately and individually, but so that really has led to this siloing of information. And so to accelerate discovery, we're trying to collate and harmonize using similar standardized behavioral measures, cognition and behavior across multiple genomic variants. And data sharing is, of course, a big part of this. So we're hoping this will really accelerate our understanding of these rare genomic disorders. So just to summarize, these rare disorders like 22q11 collectively are a major cause of developmental neuropsychiatric disorders. We know that the 22q deletion is one of the greatest known risk factors for psychosis. And the prognostic markers, things like cognitive decline and inattention, appear similar to what we see in idiopathic psychotic spectrum disorders. We see this at the level of the brain, where we see reductions in heteromodal association cortex and the hippocampus that converge with the brain regions most affected in idiopathic schizophrenia. And this does suggest that these highly penetrant copy number variants can inform our understanding of broader psychiatric disease risk. We see that a larger deletion is associated with more severe brain and cognitive impact, as well as new evidence that this 22q locus is a genomic region associated with mirrored anatomic phenotypes, a mechanism that we really don't understand and we're really trying to get to the bottom of. And lastly, just ongoing work in longitudinal and global cohorts, 22q11 and other rare genomic disorders, as well as translational studies, we hope can guide the development of more neurobiologically informed therapeutic targets. And that's certainly the goal, as well as understanding this variability in outcomes. So on that note, I will just thank all of our funding sources, and please, I'm easy to find online, so please feel free to email me with your questions if we don't get to them, but I'll stop and take questions now. Thank you.
Thank you, Dr. Bearden, for that wonderful lecture. I think that it really highlights beautifully kind of this RDoC framework that the field is really highly emphasized in the last 10 years. So I want to take a moment to encourage all of our participants to type your questions into the Q&A as we jump in. But I would like to start things off. And I think that, this framework of utilizing genetic research studies through all of the neurostructures and looking at the cortical thickness, all the way through translational studies has been really fascinating to see, as well as with the overlap with the developmental disorders. And I think one of my first question is really into, as we're doing more research in equity and health research, if there have been any kind of ethnic differences in terms of the prevalence rates across the duplications and the deletions, especially since we know that, across psychiatric disorders, there is such a difference in terms of, who gets diagnosed and when, and the severity levels. I know I'm hitting on many different topics here, but I would love to hear your thoughts, Dr. Bearden.
Yeah, no, thank you for that question. I think you're hitting on all the things that we, that I point to as, wow, we really need to do a better job of this. So yeah, I mean, at this point, there's not evidence that the 22q, that the deletion or duplication would be less prevalent in, like non-European populations, but we certainly, I think that there is, and again, there isn't empirical evidence of this, but I certainly do think that there is a delay often in diagnosis in non-European. Some of it has to do even with just recognizing facial structures. So I think this has actually gotten a little bit better now that, so in the past when, a genetic diagnosis is often made because someone would have some atypical facial features. I mean, it still is, but it's not, now more commonly, if there's a referral, say for a developmental delay, an autism spectrum, the diagnosis potentially, there will be what's called an array, a CGH array done that's sort of looking across the genome for any kind of genomic anomaly. So that's actually gotten, I think, more equitable now because that will be done across the board as opposed to looking for a particular facial feature. And because, I mean, it's just not, I think that the facial features are just not as easily recognized in a non-European person. And so I think that, some of that has gotten better. It's still certainly not like we've reached some sort of equity in terms of diagnosis, but I do think that that has improved to some extent just based on the way these diagnoses are being made. But certainly, someone still needs to get referred for a developmental delay, all of those things. We see a fair number of, at least where from Latinx populations where CHLA has a really nice pediatric genetics program, and so we do get referrals from there for our program. And so I think that and this is a long-winded answer, but it's getting better, but it's certainly not where we'd like it to be.
And so I think that one of the takeaways from your lecture, especially I think that the genetic counseling slide with the jars and balls was just particularly poignant. But would you say that for us providers or anyone that's working with families, many of us come from the developmental disability world, but really counseling families in terms of seeking out some genetic testing. I know that especially for earlier, like first time diagnosis, that is typically one of the first treatment recommendations, but is that also something that you would encourage us to do?
Absolutely. Absolutely. And I think, it's interesting because I think sometimes providers will say, oh, well, but, why does it matter if, you can't fix the genetic defect, right? But I think for many families, I mean, this diagnostic journey, having an answer to, why all of these things are associated. And I think for people with, particularly with the 22q duplication, where there does tend to be a high rate of familiality, it's autosomal dominant. So if one, it's inherited. So if a parent has it, there's 50% chance they'll pass it on to the child. And so, but sometimes, there will be no real obvious phenotype, but, it is getting that diagnosis is really important to try to put together, some of the, medical potential medical comorbidities with some of the developmental delay and other behavioral issues. So I think that it is really important for the 22q deletion. There are certain, medical management issues that also, if they're not particularly obvious, things like low calcium is really common, and, various things where it is really important to get that diagnosis early because that can really make a difference in later trajectory.
Absolutely. And I think as a clinician, I found that, even though the field is not quite at the point yet where we can have targeted treatments for, behavioral components, just the having a group that also has a similar experience where families can, join a 22q deletion group and have similar resources and go to different conferences or family fairs, I think has been one of the strongest aspects that has come out in the last, several years with the increase in genetic testing. So that's definitely a component.
I totally agree. Yeah, there's a lot of, these Facebook groups, and I think it's been just really fantastic for families to find a community that, people they can relate to. Yeah.
The Facebook groups are so true. That families are always on them, posting and just talking about the treatments and what has worked or not worked for them. So that's absolutely a huge resource. And speaking of resources, what services and other kind of interventions, things can we refer our families through CATs that are available?
Yeah, absolutely. Well, so for our 22q program, we have it's a research study, but we provide pretty comprehensive clinical evaluation and reports, which is often if it's not part of research, it's often fairly expensive or, very, very long wait list to get. So we provide that with some pretty extensive educational recommendations. There is some other web-based resources for families who are interested. That 22q11.2 is sponsored by the Children's Hospital of Philadelphia, and there's a lot of educational resources on that website. So I highly recommend that, even for if it's not 22q, but some other, either genomic or just a learning disability. They have a lot of resources there. So it's a great place to check out.
Absolutely. Yeah, thank you for that resource. And what is is that resource called again?
22q11.2.
And beautiful. I love that. That's very sweet.
Yeah, exactly. And and so if you want, I'm happy to provide other sorts of links or that kind of thing. But if anyone's interested in participating in our study at UCLA, please feel free to just send me an email, and we'll be happy to discuss and or schedule an evaluation.
Absolutely. I think that it's such a wonderful resource to have families be able to come through under research where, like you said, these full scale assessments and evaluations can be so lengthy as well as costly. So to be able to offer that to families is really wonderful.
Yeah, and we have a lot of incredible trainees that work in our program that just do a great job. And, even we see a lot of families that are from out of state. We're doing, most of our assessments are virtual these days. So, but we're able to provide, even if it's not a local recommendation, we try to find referrals that are appropriate for folks that are out of state as well.
Wonderful. Excellent. And so is the best way to contact CAPS through their website or directly through you?
Yeah, you can email me. You can email CAPS. CAPS is caps.ucla.org. Yeah. So maybe I could, if I can put it in the chat right now. But yeah. So if you look up UCLA, just Google CAPS UCLA, and there's a way to put questions. You can directly, you can put your contact information on the website.
That's great.
Confidentially, and we can get back to you.
Okay, excellent. Well, thank you so much, Dr. Bearden, for this excellent talk and for taking the time on a Monday during the holiday season to be here with us, providing resources and letting us know what's happening at CAPS.
My pleasure. Well, thank you so much for inviting me. And yeah, like I said, I'm always happy to hear from folks at any point. So, please don't hesitate to get in touch.
Thank you. Well, and last minute plug for all of our participants to make sure to fill out your Tarjan evaluations. It's in the chat. I'm also seeing in the chat lots of sentiments of gratitude. So thank you again, Dr. Bearden, for this wonderful talk and for educating us on 22q.
Thank you so much. And thank you all again. And happy holidays to everybody.
Happy holidays, everyone.